This method can help answer key questions in the field of genomics and gene regulation, such as how multiple gene regulatory regions, also known as enhancers, work together to control transcription. The main advantage of this technique is that multiple enhancers near multiple different genes can be tested simultaneously in order to rapidly identify regions that are involved in gene regulation.
To begin guide RNA design, first generate DNA sequences as described in the text protocol. Use a program such as eCRISP on the generated DNA sequences to find guide RNAs with low off-targets. Guide RNAs consist of 20 nucleotides upstream of a protospacer adjacent motif, which takes the form of NGG for the dCas9 from S. pyogenes. On the eCRISP website, select the organism of interest using the drop-down menu. The genome assembly appears to the right of the species name. Select the Input is FASTA sequence radio button. Copy the FASTA sequences from above and paste them into the dialog box. Ensure that a FASTA header is included for each sequence. Select the Medium radio button and Single Design in the drop-down menu. Click the button Start Single Guide RNA Search. A new browser tab will open and results will be displayed. Download the candidate sequences by clicking the button Download an Excel-formatted tabular report for all query sequences together. Next, use the UCSC genome browser to blat candidate full-length guide RNA sequences to the genome. In a browser, navigate to the UCSC genome browser website.